well, what do we spend the money on? I've kind of arranged these in order of things that don't cost so much and things that cost more. So, is there a pointer on this?
Yes.
Sorry.
Right here.
Oh, okay. So these are small pieces of equipment in the $100 to $1,000 range. When we have, half a dozen of these things. These are things that run $1,000 to $5,000 in their range. We have about a half dozen of these things. These are centrifuges. These are things that separate DNA. You talk about recombinant DNA and genes. Well, that's how we actually handle them. One of the ways we handle them. When we have patient samples or cells that we grow, leukemia cells that we study, we have to keep them frozen when we're not working with them. And so this is a liquid nitrogen freezer. I don't know if you've ever seen movies like Jurassic Park, when that guy stole the embryos and had that little thermos. That was liquid nitrogen, and this is you couldn't carry this around in your lunchbox. But I mean, this holds maybe 50,000 vials of tumor cells, and this little unit here costs about $40,000 to buy, and it costs about $7,000 a year to feed liquid nitrogen to keep the cells frozen. So every lab group has to have these. These are machines that take small amounts of DNA and make them into big amounts of DNA so you can study them. These machines run anywhere from $20,000 to $100,000. This is actually a big bargain. This is not what this is growing cells. We grow leukemia cells in dishes and we do experiments on them. We inject them into mice. This is actually a big bargain. This doesn't cost much. It's maybe on the order of any experiment will cost a couple $100 to $1,000 to run. But this is actually where a lot of creativity comes in the process. There's kind of two aspects to science. There's the non-thinking data gathering phase, and things like collecting, demographic information, mutational data. These are all data gathering. It's kind of relates to that big data thing, that places like Google do. They collect mass amounts of data, and that's the data gathering phase. And then you have to kind of look at that data and think, are there patterns in that data that allow me to understand what's going on? But if you have an idea just from descriptive, looking at associations in data, that doesn't prove anything. It's an association. There's this classic phrase you might have recalled from university that association does not imply causality. Just because two things are related doesn't mean one causes the other, or that they occur together. So this is where a lot of creativity comes into science, to actually design an experiment where you can make a variation and determine whether or not that variation caused something. And simplistically, does this gene cause leukemia? Because you can have lots of genes. Lots of genes are mutated in leukemia samples. That doesn't mean that they all cause leukemia. They just happen. Some are there along for the ride. They're called passenger mutations. But it can take a lot of work to design a good experiment to ask these questions. And so here we have a graduate student and an undergraduate student who, I just took these pictures at 5:30. So these are students right before coming here. These are people, they work into the hours, they skip dinner, they come back at night. So this is the kind of dedication that you get for your dollars. People are working all night, all weekend. They commit their lives to it because they really are passionate about it. So I think that's one of the values you get from scientific research, people who really care about doing the work. But again, they're basically growing leukemia cells in these dishes, looking at them under microscopes and growing them. But this is not very expensive. This little room here behind the glass, there are dangerous viruses behind that wall. It's just we work with biohazardous materials in order to make cancer. And at the end, I'll show you, we can actually make leukemia in a dish now. This is our big advancement that is not published yet. But we can make leukemia in human cells. But for me or my trainees, to take these elements in that little tube there, for example, and put it into a cell and make it into a leukemia cell, you also have to realize these people are taking personal risks. I mean, they're working with things that can cause leukemia. And so This is not dangerous, is why they're not, she's not wearing a lab coat. She should be. But those stuffs aren't as dangerous as ones. When they actually do the really dangerous stuff, they have to put on, many more layers of protective equipment and go into this special room, which has, special ventilation and anti-chamber door locks and all these kinds of things. This is kind of the more expensive range of things. When we look at leukemia, is kind of one of the things people do these days. Instead of looking at, when you go and get a biopsy, and you think, well, someone's going to look at my cells, right? And you might think, well, in order to look at them, even under a microscope, you probably look at on the order of 1 million cells, tens of millions of cells at a time. And that's really how, as a pathologist, for example, I would diagnose a disease. My eyes glance over, tens of millions, hundreds of millions of cells before I can say this is an AML, or this is an ALL, or this is MDS. Maybe it's going to be AML. So, but in order for science has kind of moved to the point where, partly because technology allows it, we don't really look at cells 1 million at a time. We look at cells 1 at a time, every cell. So if there's 1 million cells, we look at all 1 million of them. And so these machines, these are called flow cytometers that help you do that. These machines are about $500,000 each. This machine is about $800,000. This machine is $1 million. And there's an older machine that, anyways, I don't know the cost of that. I wasn't around when we bought it. But anyways, these are not my own machines. When you have machines of this sort, groups of investigators combine and they share them. And so these machines are all shared by the entire building of 50 researchers at the BCCA across the street. But I just wanted to give you an idea. This is the fanciest of technology that we do these days in terms of single cell analysis. And so it's just the evolution of technology has allowed us to ask deeper and deeper questions. And obviously with 1 million cells, if you look at them 1 at a time, in some sense you get 1 million times more data than if you looked at all the cells together. And so that's kind of that issue of how much data we have to deal with, the information that people have to handle, and how do you organize all that in your mind and come up with a singular thought that allows you to come up with the next experiment.